Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This is the third one in the protein series. It's probably a bit less important than the first two, but this is far and away the most important playlist to watch. This one, though, is very, very important in that it's a topic that comes up a lot in exams. So if you want to score highly in the exams, this is a really important video because this talks about factors that affect enzymes, which means you have to apply your knowledge of how proteins form, what they are, how enzymes are, what the mechanism is for how they break down their substrates. So this is very important from an exam skills point of view, but the fundamental concepts were covered in the first two videos. It's still GCSE level, and please do subscribe to my channel if you haven't done so already. Thanks. So let's just quickly recap enzyme formation, but we're going to do this really quickly because remember, they're still just proteins. So the mechanism is exactly the same as the other proteins we've looked at. You've got a ribosome that reads the DNA. The DNA is just instructions for the sequence of amino acids. That chain of amino acids is called a polypeptide. Polypeptide because peptide refers to the bond in between them, what holds them together. Poly meaning many, so it's many bonds, polypeptide. Okay, that polypeptide chain has a very, very specific sequence though, and of course that's what was coded for by the DNA, by the document up there. Okay, if we shrink that down, if you remember from what we did last time as well, you've got many, many amino acids that would make up the polypeptide. So let's just substitute that for a long blue line. So that long blue line is the chain of amino acids, it's the polypeptide. Now, to form an enzyme, of course, they're going to fold in their very specific way, and that specific folding is because of the sequence of amino acids. We looked at it in one of the last videos. And if we just draw around the outside of that to give us the more familiar picture of an enzyme, we can see that you've got the active site, which is the mouth part, and it's the mouth part that does the work for the enzyme. It's kind of the equivalent of your hands. That's where it grabs hold of stuff, builds stuff, or breaks stuff down. Remember, that depends whether it's a catabolic enzyme or an anabolic enzyme. So the active site is where the building or breaking molecules occurs. So that's a really important kind of run back through. What we need to focus on now is what factors affect how well enzymes can work. And if you remember the mechanism, it's an enzyme that will lock together with a substrate that is complementary and turn that substrate into products. It could be one substrate into multiple products or multiple substrates into one product. Okay, again, depending on whether they're catabolic or anabolic. So let's just explore those main factors. So the first one, and the really important one, is temperature. This comes up every single year in the exams. I cannot stress how important it is that you learn this really well. It might just be that you get one standard question of explain the effect of temperature on enzymes. We'll answer that in the next slide. I'll give you a good exam answer for that. Or it could be that they give you data from an experiment and they ask you to interpret a graph or explain what's happening on a certain bit of the graph, but it's always in the exams. And over the course of an entire question, it can be worth up to sort of 7, 8, 9 marks just on this because it links up to protein formation and enzyme function. So let's have a look at this. We're going to put this as a graph. So we've got temperature on the bottom axis, on the x-axis. That's because that's the independent variable. The dependent variable is enzyme activity over on the y-axis. So if we just fill that in, we get a pattern that looks like this. Okay, as temperature goes up, enzyme activity increases. Now that is all due to collision theory. Now you should have done collision theory in your lessons. You almost certainly will have done it in chemistry, but collision theory is basically about the following points. Okay, and all I'm going to do, by the way, on this slide, I'm going to give you the key points that explain what's happening with this. On the next slide, we'll put that into a decent exam answer. But collision theory is basically when you add heat. Heat is a form of energy. If you're giving energy to the particles, the substrates and the enzymes, then there's going to be more movement. That thermal energy will be transferred to kinetic energy, so they'll just move faster and faster. Now, if they're moving faster and faster, there's going to be more collisions. Collisions as in crashing into one another. Now, those collisions, though, will only be successful if they're between the active site and the substrate. So if there's my enzyme and there's my substrate, the collision must be like that, so that the substrate goes into the active site. If the collision were like that, well, it's not actually going into the enzyme. But of course, the more overall collisions you've had, if let's say 5% of collisions are that, and the other 95 are all that, if you increase the overall number, then the number of successful ones is still going to get much larger. So by giving the particles more energy, more heat, they can move more, collide more. When the collision is between the active site and the substrate, they'll form enzyme substrate complexes, which means you're going to get products. And the production of products is the defining thing about enzyme activity. Okay, so the more heat you add, the faster they move, the faster the reaction. However, that's only half the answer. So we'll call that part A. So I've labelled on the diagram there, you can see along the bottom, that's A. I've put an A on the graph. Because there is a B, there's a second half to this graph, which if I reveal it, it crashes straight down. That line doesn't just keep going up and up and up and up, like you can see on the graph. Yes, the more heat you give the particles, the faster and faster and faster they move. However, what's happening at B is something called denaturing. Denaturing is basically melting. It's when the enzyme that's shaped like that should be hitting like that, and with more and more heat, that's happening faster and faster and faster. But as it gets too hot, the enzyme literally starts to melt and lose its shape. Okay, that means there is no longer an active site for the substrate to react with. Okay, let's put that in a few more words though. What we mean when we say we're melting, we mean that the polypeptide chain that we saw on the previous slide is starting to unfold. So that folded structure that gave you the enzyme, if that chain unfolds, goes back to being that straight blue line, obviously doesn't have a shape anymore. And remember, shape determines function. That's why I've put shape up here, look in bold, in capital letters. This is the important bit about denaturing. You lose the shape of the active site. Without the correct shape of the active site, it's not going to be complementary to the substrate anymore, so you're not going to get any products. Okay. Hopefully that makes sense, because this is the shape of the graph that you always get. Now, it could be that they show it on the side as enzyme activity, or they could show it as amount of product formed, or they could show it as the level of substrate disappearing. Doesn't matter, they all come back to the enzyme rate. So it's going to be a graph similar to this, where you're going to get a peak and it's going to drop down either sides. Now that peak is important though. If I draw a line across the top of the graph there, that you can see is the maximum activity that the enzyme gets to. And we can draw a line down to the x-axis there, because that's called the optimum temperature. That's when the particles are moving as fast as they can, but before the enzyme has melted, has denatured and lost its shape. After the optimum, there's still more and more collisions because they're moving faster and faster. But now those collisions are pointless because the active site has lost its shape. Now, because this is so essential, that comes up every year in the exams, we're just going to take a minute to build up an answer to the question, how does temperature affect enzyme activity? So I'll put the graph back up there again. This is the kind of answer I would like one of my students to give. This is probably way beyond what you'd need to give in an exam, but it gives you an idea of the level of detail that is expected. Okay, so question, how does temperature affect enzyme activity? Answer, part 1. With increased heat, and I put all the key terms that could be worth marks in the exam in capitals, and I've coloured them gold. Okay, so with increased heat, the enzymes and substrates have more energy, so they move more and have more collisions. If the substrate collides with an active site, they can form an enzyme substrate complex and turn the substrate into products. And as the heat increases, so do the collisions, so the enzyme activity also increases. That's 7 points. And we're only on side A, that first side, okay? A middle bit, the rate of reaction will keep getting faster and faster until it reaches the optimum temperature. So we've now got an 8th mark by including optimum temperature there. Moving on to side B then, as heat continues to increase, it destroys the bonds. Bonds is quite often worth a mark in the exam. That holds the shape is essential, of the enzymes together. The enzyme is denatured. Denatured is the most important term here. If you were trying to prioritize things, side A is about energy, side B is about shape, okay, and denaturing, which changes the shape. Those are your priority things. So denatures, which means the polypeptide chain of the amino acids has begun to unfold. It's unfolding, the reverse of the folding that formed it, which changes the shape of the active site. I've lost count. So it's no longer complementary to the substrate. Even as collisions continue to increase, no enzyme substrate complexes can form, so no product is made, and the rate of reaction drops rapidly. What's that, like 15, 16 points there? And look at what I've written. It's not actually a lot written here. It's a couple of paragraphs, okay? The key thing is, look how scattered it is with keywords. I can't tell you how many times I've marked exam papers where a student's given me what is kind of a correct answer, but I've hardly been able to give it any marks because it doesn't have detail, it doesn't have key terms. They might say something like, reactivity increases as it gets hotter, and that gets to a peak and then it goes down afterwards because the enzyme changes. There's nothing inherently wrong with what I've just said. It is correct. There's not a single key word. There was no mention of movements, of collisions, of active site, of energy, of denaturing. So just be careful. If your answer doesn't include detail, it's not going to get any marks. Just one last thing I want to put. I'm going to insert something in here. I didn't want to put it straight away because I wanted to move on. But now that we've written that last paragraph, we can go back in and fill something in to clarify what we meant by the optimum temperature. So you'll see I put a line just here. The optimum temperature is when the collisions are happening very rapidly, but before the active site loses its shape. Now, there's no key terms in there, so in itself it may not be worth a mark, but it does add detail and it adds explanation to what you're showing. It demonstrates knowledge, which examiners love, and it means they'll be reading it and thinking, yeah, that's really good. And if they've got brackets, like they can award either 1 or 2 or 3 or 4 or 5 or 6 based on your demonstration of knowledge, they're definitely going to give you top marks if you've qualified every statement. So let's move on from temperature and look at concentration. There's only 3 of these, by the way. There's temperature, concentration, and then we'll move on to pH in a minute, and that's it. Okay, but concentration. Let's do this by an analogy. Let's give you a couple of situations. So situation A, I'm going to put 2 people into this box. Okay, we've got the guy in green, let's call him Richard. We've got a guy in blue, let's call him Julian. So Richard and Julian are inside this container. They can't get out of it. That's the only space they've got to move around. Now Richard, on his hand, has got a big smear of oil, dirty, greasy oil, and Richard's a bit cantankerous. And anytime Richard bumps into anyone, he just smears his hand down their face to smear them with oil. Now, Julian doesn't want to get smeared with oil, but Julian can't avoid Richard. But also, Richard can't actively kind of hunt Julian, because I need you to imagine in this space, they're both blindfolded, they've both got earplugs in, they do not know where the other one is. All they can do is wander around within that space. If they bump into a wall, they just bounce off and go in the opposite direction. Now, with only the 2 of them in there, the chance of Richard bumping into Julian and being able to smear him with oil is pretty low. If we create situation B though, and in this same container, look, it's exactly the same size. If I put Richard back in there with his oil on his hand, but this time in situation B, there's a whole load of Julians. They're everywhere. There's 10 of them filling the space that is in there. Now, even if they still had their blindfolds and their earplugs in, if they were all just wandering around, Richard's going to bump into a Julian really quickly, smear them with oil. He's going to turn around, he's probably going to bump into another one really quickly, smear them with oil. Almost every time Richard turns, he's going to bump into a Julian and be able to smear them with oil. Okay, now I'm going to throw up a third one here. This time in situation C, I'm still going to have just 1 of Richard and 1 of Julian, but I'm going to put them in a smaller area now. Now, hopefully you should be able to look at that and compare it with what happened in situation A and situation B. In C, whilst you've only got the same number of people, because they're in less space, you've increased the chance of them bumping into each other. You've increased the chance of Richard being able to wipe oil into Julian's face. Okay, what we've done in situation C, technically we've increased the concentration, even though we haven't increased the number of people. All we've done is reduce their space. We've put them under pressure. And the key thing to remember, that increasing pressure is basically the same as increasing concentration. Now, how is this relevant to enzymes? Hopefully that's really obvious to you. Richard in these scenarios is the enzyme, the one looking for the other things. The Julians are the substrates. So you're trying to get Richard to the substrates, so his hand, acting as an active site, could lock onto them. The chance of those collisions is greatest here in situation B, because the number, the concentration has increased. If you also increase the pressure, you reduce the space further, you're going to further increase the number of reactions, the number of smears that can happen. So if you put more things in a smaller space, that's two variables there, the number of things and the space, they're more likely to bump into each other, which, as we know from the previous explanations on temperature, we call collisions. Okay, more collisions equals more reactions. So enzyme activity is fastest in higher concentrations or pressures. That should be pretty obvious. Okay, last one, and we'll go pretty quickly on this one because this is really simple to understand. I'm putting this up again just because it's really, really relevant to this. The ribosome reads the DNA, puts the amino acids in sequence. That sequence of amino acids folds in a very, very specific way to create our enzyme. And again, if I draw over the outside of that and label it up, there's the active site, there's the substrate that fits or is complementary to that active site. Now those two can fit together, and the enzyme folded into that specific shape with the specific function of breaking down that substrate. It folded into that shape because of the sequence that was laid out in the DNA that's up there. Okay, now there's a qualifier to that. It folded into that specific sequence, into that specific shape, only when it's in the correct pH. If you put it in an incorrect pH, it will fold into a different shape. Look at what I've got right next to me here, look. If that same polypeptide, look, it's the same blue line. If we took that same polypeptide, put it in an incorrect pH, it will fold up to a different shape. If I draw around the outside of this one now, right next to me, look, you can see it's very, very obvious. The active site is totally different in shape to the active site that we want on the enzyme over there. So that substrate will not fit into this enzyme, even though it's made from exactly the same polypeptide, same sequence, coded for by the same bit of DNA, made by the same ribosome. Doesn't matter. If it wasn't in its correct pH, it won't form its correct shape, which means it won't be able to perform its function. So the key thing there is enzymes will only form their correctly shaped active site when they're in their optimum pH. Now that means you get a graph quite similar to the temperature one. In the enzyme activity is very low in the wrong pH. Soon as the enzyme's in its right pH, lots of activity. If it goes too far the other side of the pH, it goes down again. Okay, it's quite straightforward, but it's a very standard, practical, very standard investigation that comes up quite a lot in the exams. You can also link this up to the digestion stuff because there's a protease enzyme that acts in your stomach. Not really any other enzymes work in your stomach because your stomach is full of stomach acid. Protease is one of the only enzymes that you have that forms its correct shape in acid pH. If you put any of your other enzymes in the acid of the stomach, they'll form their wrong shapes and they won't work. It's why you have to release bile to neutralize the acid to let the other enzymes work in your system. But we'll do more of that when we come to the digestion lessons. Now, I'm not doing a summary slide on this one because we have three very different areas there. And to try and summarize all three in one would just get too complex. But please remember, temperature, concentration, and pH have very significant impacts on how well an enzyme can function. You are likely to get questions on any or all of those three factors. So make sure you've gone back and reviewed all three and you know exactly how to describe those effects. From here, go on to digestion 1, which is an introduction and process overview. That looks at what digestion is and why it's so important. The digestion 2 lesson then focuses a bit more on key parts of the digestive system and how they've adapted to maximize their function. As I said at the start, please do subscribe to my channel if you haven't done so already. Like this video if it was useful, and visit my Facebook page for some more weird off-piste biological facts just to keep you interested. Thanks a lot.